calcium pectate. Proteins, next chapter. Proteins are the most important organ, and it is the substance known so far in the living cells. The name protein was suggested by Berzelius, the common questions, in the year of 1938. It is derived from Greek word proteos, that means first rank. As per our nutrition, protein is very, very mandatory things. So we should give the preference of nutritional values for proteins. That's why this is under the category of first one rankers. Proteins are the polymer of amino acid where each of the amino acid is linked with another by peptide linkage. This is the difference between carbohydrates and proteins. In case of carbohydrates, this is the glycosidic linkage, but in case of proteins, this is the peptide linkage. Okay. And if we go the structures of the proteins, there are quite differences between the linkage group has been changed sometimes, and the complexity is getting more and more functional. Amino acid is the basic unit of protein. So out of 20 amino acids present in the nature, combine each other to form a specific proteins. So that means a number of amino acids are linked by peptide linkage and make a common polypeptide chain like this. Okay. So cysteine and hydroxyproline are the most commonly found major proteins. Okay, this is the questions also come. And the simplest amino acid is glycine. This is also another important questions. Structure of proteins. There are 4 different structures has been found in the natures. They are primary, secondary, tertiary and quaternary. I'll explain each and every one very, very carefully. You have to be understand. First one is primary. You can see here the linear sequence of amino acids joined together by peptide bonds is termed as a primary structure. The primary structure is like this. Single linear structure of amino acids are attached to each other by peptide bonds. Only peptide bonds, peptide linkage is present. Okay, peptide linkage is formed by amino group and the carboxyl groups. Amino group of the first amino acids and carboxyl group of the second amino acids. Amino group of the second amino acids, carboxyl group of the third amino acid. In this way, one by one, it has been attached. We know the amino group. What is amino group? That is NH2. This is the, suppose this is the amino group of the first amino acids. Now it will be attached with the COOH carboxyl groups of the second amino acids. So here H2O will be released. This H2 and one oxygen has been released to make this amino glycosidic peptide linkage. So structural proteins, the primary structure, the linear arrangement of amino acids include covalent disulfide bonds between cysteine residues. Sometimes it is involved in the making the disulfide bonds between the cysteines. In case of cysteine amino acids, there are 2 sulfates groups are attached to each other where amino acid linkage are grouped by themselves. One by one it has been side by side gone. The examples, first one is myoglobin, a globular protein made up of a single polypeptide chains of 153 amino acids that is folded into 8 alpha helix. So myoglobin is a very, very easy example. Another is lysozyme. Lysozyme and myoglobins, these are the very, very important examples. It can come into your the questions. So hemoglobin we can say, but hemoglobin is a part of, there is a part of alpha helix chain and beta sheeted chain. Each and every alpha helix chain can be considered as primary structure. Okay, so next one is hemoglobin. So hemoglobin, in case of this is present in our bloodstream. What it is from? Hem means one iron content is there, and another with it we have the globin proteins. We have the globin proteins. Globin. So in this is a one type of conjugated proteins, and this is having the structure of secondary structure also and primary structure also. So primary structure is the simplest structure where the amino acids are attached to each other by peptide linkage. Cytochrome is another examples. It has a cofactors. Now come into the secondary structure. How secondary structure can be defined? Secondary structure also have two different types, alpha helix and beta sheets. So alpha helix and beta, how we can define? Suppose this is the secondary structure like this. This suppose this one. So this is the helical structure of amino acids where this is dot, this dot. Suppose just change the color so that you can easily, suppose these are the red points indicates the amino acids. So each and every amino acids are linked to each other by peptide bonds, and it will make this helical structure. So now the interaction between these two molecules, of amino, suppose this one and this one, how they will be interacted? They will be interacted by bonding by hydrogen bonds. So there are two different bonds that involved like this. Here it can be linked to each other, here and here. In this way they have been making the stability of the helical structures stable. That's why this structure is stable. So there are two types of bonding. Amino acid to amino acids parallelly it is a peptide bond, and vertically they are attached by hydrogen bonds. So another structure is, suppose this is the first chain of the polypeptides, this is second chain of the polypeptides, and if we give the colors here like this, and here like this, if any type of linkage has been found between two peptide chains from one amino acid to another amino acids, that may be hydrogen bonds, this type of structure will call beta plated sheet structure. Sheet structure. This is very important for your exams, and this is the alpha helix structure. So coming to the next point, secondary structure. The two most common types of secondary structures, alpha helix and beta plated sheet. You can easily remember: myosin containing two heavy polypeptide chains and two pairs of light chain arranged in globular form. So myosin have two alpha helix and two beta sheet structure. And next is tertiary structure. This is again very complex. So primary structure containing proteins are not stable at all. There are very few examples of proteins that are stable and functional, like lysozymes, but most of them cases the tertiary structure is stable and functional because a specific functional units or specific structure is there. So how, suppose this one is the first chain, this is the second chain, and this is the third chains are close to each other in like this way. Suppose this is the terminal ends where amino group is there. What it is? Amino group. And this is the another chain where the terminal end contain COOH or any type of other group, suppose COOH. If the bonding is possible, bonding is forming by sharing a common substance, bridging them, get together a common functional to form a common functional unit. Suppose these particular terminal ends of amino groups and carboxyl groups are shared by a single common substances to make this particular area is functional. In this way, this is can be possible by these two terminal ends of these two chains again for this. So there may be a different chances of this case. Okay, so in this way, this is the typical structure, typical complex structure of tertiary structure of proteins. It refers to the arrangement of secondary structures into three-dimensional structure. Three-dimensional because in the three ways the chains are, three different chains are attached to each other and make a common bonds to each other. Native conformation is maintained by multiple non-covalent bonds. So native conformations will be, should be non-covalent bonds. It makes the protein functional. This is the important things. Up to secondary structure, the protein may or may not be functional. From the tertiary structure, which should be functional. Myoglobin of muscle is the example of tertiary structure. So the next one is quaternary structure, the structure produced by association of more than one polypeptides. So what is the chances? Suppose in this case, suppose this is the this is the first chain, this is the second chain. If another set of chains like this and like this, so these two chains may be alpha helix, alpha chain, alpha chain, and these two are red color chains are beta chain. So they will form a complex structure. So okay, another thing is that most of the enzymes are under the category of tertiary structure. Enzymes, most of the enzymes, you have to remember, all the enzymes, most of the enzymes are under tertiary structure. Okay, so example is hemoglobin for quaternary structure. You just see this picture diagram. This is the primary, secondary, alpha helix. C is the tertiary, means three different chains are meet to each other. Quaternary means a complex structure of two different types of chains are conjugated or attached to each other by covalent bonds or any other bonds. So classification proteins, simple proteins contain only ordinary alpha amino acids. The very important examples are albumins. The questions are coming. Globular proteins, the most common serum albumins are the examples of simple proteins. Globulin also another types of simple protein. Example, family of globular proteins that have higher molecular weights than albumin. Albumin is present in egg white. We know that. Each and every one we know that. Glutenin, it is the basically sticky like substances, produce sticky like substances in most of the stored grain crops, and glutenin content crop is wheat, rice, everything. Protein aggregates that are the major proteins within wheat flour. You can see the softness of the wheats come after mulching that particular wheat powders with water so many times, so that this particular stickiness are coming. Prolamine is also another examples of simple proteins. So histones, another one. Histone is basically consisting of nuclei that package and order DNA into structural units called nucleosomes. So histones is the proteins where histone is present, as well as it has non-protein component is also present, non-histone components. Conjugated proteins, this is the complex structure. Proteins that contain non-amino acid components in addition to amino acids. So essential amino acids are there as well as non-amino acids. That means no such amino acids are present. Amino acids is the primary structure content polypeptides. With this, non-amino acids containing any types of substances can be attached to each other. Example is nucleoprotein. You just can see the example. Protein part is there and nucleic acid is attached to here. This is the non-amino acid part that's form the chromosomes. Another example is glycoprotein. Protein part is there and glycogens attached to each other as a sugar unit to make these conjugated proteins. Lipoprotein, lipid proteins is lipoprotein. Metalloproteins is the very good examples of metalloproteins. Metal plus proteins. Derived proteins. Another types, degradation products obtained by hydrolysis of natural proteins with acids, alkalis and enzymes. So any types of proteins that is hydrolyzed by natural ways with acids or alkaline and make the derived proteins. That's why this is the derivatives. Example is peptones, peptides, metaproteins and coagulated proteins. All are the examples of derived proteins. Various structural proteins, these are the examples you have to remember. Structural proteins, collagen, muscle proteins, keratin in hair, wool and nail, where it is situated, elastin found in insect veins. So all the examples are very important. Myosin, major proteins in muscles, usins found in the bones and teeth. The questions are coming from this, I said JRF from here. And melanin, protein of skins, hairs and colored layer of the eyes. All enzymes are the, so these are the very common points you have to be just remember. The name of the proteins were suggested by Jan Jacob Berzelius. This is also important. Composition of proteins, these are the key points I have mentioned. You have to just remember molecular weight of the protein should be in between 10 to the 4 to 10 to the 7. Legumine is a very good examples of pea, and tuberine is a good examples of potatoes, are the common examples of globular proteins. Prolamins, again this is the very important protein. It is found in this is all insoluble because this is not have hydroxylic group. There is no such hydrolysis can be possible because hydrophobic attachments are there. Only known in the combination of nucleoprotein of sperm of fishes. Glutenin. Glutenin is also another type of insoluble globular protein, conjugated protein. Gliadin in wheat, hordein in barley and zein in maize. These are very good examples of plant proteins, and so many questions are coming. Histone proteins are the soluble in water. The questions is also coming. That's why we can see in our muscle cells, in our chromosome, the histone proteins is easily soluble into the bloodstream and used for any types of cell divisional functions. Nucleoproteins and glycoproteins, another